So now we're starting. Now we're going to talk about how to actually compare genomes. And right now, like I said, we're beginning to be able to sequence other things, but historically the bacteria were the first genomes you could really start to compare. So, and we'll be talking some about atlases, and that's what these things are, little wheel plots looking at it. So this is our regal cat. This is Bob the cat because he's got a little white smudge in his nose, and he's got a little curl in his whiskers. That was kind of interesting. He curls his mustache. Okay, so we are here comparing bacterial genomes, and actually, oops, sorry, I took that extra slide out. Okay, never mind. So what I've done is tomorrow I want to talk about viral genomics and human genome project. But what I might do is take this lecture, Introduction to Atlases, and move it to tomorrow because we already spent 45 minutes. I'm supposed to be done at 9:15 and it's 9:13 right now. That's okay. So probably what we'll do is but I would like to talk about proteome comparisons as well. And so what I might do is talk about atlases tomorrow. We'll see. Or yeah, but okay. So I've purposely shown you guys these web pages where NCBI, where they have all the genomes. And so you can go to those pages. And in fact, what I can do is, what I'm going to do is this. I'm going to go back. Let me stop sharing for a second. And I'm going to go back to my browser. And go here. And I'm going to go into chat. So I paste a link in chat. Can you guys see that?
We are good, yes.
And in that link, you can use that link to answer these questions. Largest bacterial genome in GenBank. What is it? And how big is it? Can someone do that? You guys are very shy this morning. Haley, I know you're not shy. What's the largest bacterial genome in GenBank? Actually, is Haley on there today? She's not. Well, Andrew's usually very talkative. Andrew?
Sorry, I'm running around right now. Once I get back to my computer.
Okay.
Tending to my plants.
Okay.
Fine.
So anybody want to answer? There's a web link that I sent you guys. Go to that web link and you can sort. So you just say, just show the bacteria and sort by the largest. Well, if no one's going to answer, what happened here? Why? Ah, that's interesting.
Is it 35 MB?
What is the name of the organism?
Oh, sorry.
The size sounds right.
Just says bacterium.
It's a bacteria. All it says is bacteria, right? That's not very useful. Okay.
Right. Yeah.
Okay. So that answers that question. We'll talk about this in a minute. Okay. Now, if you click on that link again, Juanita, and say, now what's the smallest? What do you get?
Candidatus parawarcaeum.
Candidatus something? And how big is that?
0.100212.
0.100. So that is 0.1 thousand bases, right? So that is 100,000. It's 0.1 megabases, so it's 100,000 bases, right?
Mhm.
That's more or less, that sounds reasonable. That might be right. The other one wasn't 35 megabases is too big, but okay. But that, I mean, that's what's there. So that's the correct answer. Okay, any idea, E. coli, how big should E. coli be? You guys
5
by now,
megabases. I mean,
it's around,
should be
it should be
around
around
5, like
5.
4.8.
And it's basically 5 plus or minus 1. So some of them are 4 and some of them are 6, but it's in that range, right? Around between 4 and 5. Okay, that's what it should be. And then now, again, go to that page and how many bacterial genomes are complete? You guys know how to do that. So let's go here. Stop sharing again.
Only bacteria, no archaea.
Well, that's the other question. So by bacteria, do you want eukaryotes? So sure, just tell me bacteria for now if you can.
6,237.
Does that sound right? It might be.
I excluded the partial.
And we go here. And so we say we're going to do bacteria, right?
Oh,
And
454.
But then, hang on. Shouldn't I be able to do other things in the filter? Now I'm confused.
Yeah, your filter looks smaller.
Wonder why that is. So this is everything. There we go. So now I say, I want that's complete and that has chromosomes. It turns out that those are kind of two different, they mean the same thing more or less, that are finished. So when you do that, you wind up with 27,000, it says. Does that sound right? Yeah, 23 and 24. So it's 27,000. Now you say how many bacteria here. Yeah, and that's right, because it gives you those numbers there. So there's only 454 archaea that are complete, and there's 26,000 bacteria that are complete. Okay.
But then what does it mean by the list that they actually give you? So this list doesn't, because there's only 5, oh, never mind. It does say 26,000 on the right-hand side. I was looking at the middle column, sorry.
Yeah, it says 550 pages. That's what's
Yeah.
confusing. Yeah, okay.
Got it.
So let me go back over here and stop sharing. So you guys, in a way, congratulations, we finished the, you've answered all the questions now. So, okay, that's the end of the lecture. Well, kind of. So here's the 4 questions that we have. What fraction of genomes are draft? Well, if you figure out the ones that are complete, you just subtract and those are draft, right? So my question is 10% of the genomes complete? Is it more than 10% or less than 10%? You should be able to figure this out, Andrew. You said 27,000 out of how many? 300,000?
Yeah.
I believe less than.
So how many are draft? If they're not complete, they're draft. So it's more than 90% are draft. Okay. So you can see working with this data is a little bit sometimes, trying to get the right word and one of the things is there's just been this real growth, 300,000 genomes. I mean, when I first started this, well, actually, yeah, I got an email from a student, and I'd forgotten about this. This is in 1994. I was doing my postdoc at Oxford, and I had a PhD student I was kind of co-supervising. And his boss was one of the world's expert on Haemophilus influenza. And this guy from the US came and gave a talk and about Haemophilus influenza. And he said, oh, by the way, here's the entire sequence of this genome. This is a year before it was published. And so my student said, what can I do with this? We have a sequence, for a genome. This is the first bacterial genome to be sequenced. And we played around with it and kind of came up with a lot of things, but that's kind of how I got into it. And then in 1995, 2 genomes were published. In 96, there are 4. And now you see there's more than 300,000 genomes published. And it's just, there's almost 1 million SARS coronavirus genomes sequenced. And so what's happened is that we've just been growing exponentially. When I talk about the Human Genome Project, I'll talk a little bit about what's behind that. But the bottom line is that, and so here, this paper was written in 2015. So this paper is 6 years old, stops here. And it's saying, okay, here's where, back in 2000, when they were doing Sanger sequencing, it's called first generation. And with second generation sequencing, it starts kind of ramping up. And now that was 2015, and they're kind of projecting, and they give 3 projections here of how fast things are going to go in the future. And it turns out now in 2020, we're here, we can actually see how good are these 3 predictions. Well, we're actually producing more than they thought. And if you look, it's not that far. It's more or less going along the same lines as before, where it's growing really, really fast. 2 zettabytes were sequenced last year. 2 zettabytes, 2,000 exabytes. That's a lot of data. And I'm going to talk more in the Human Genome Project about this, but basically a petabase is 10 to the 15th bases. So if you look at the scale, that's 5 times 10 to the 15th, 10, 15, 20. And you can see basically until around 2012, you don't see anything on here. The GenBank is growing. So this scale is 1 petabase. So that's 1 unit is that much DNA. And you have to kind of look at this. So because, so a petabase is 10 to the 15th and exabase is 10 to the 18th. And I showed you that earlier slide. Zetabase, that's 10 to 21. So this is an awful lot of data. So this is second generation sequencing, and we're here, what's called third generation sequencing, portable things. And so that's just a lot of data, bottom line. And so you can kind of watch timeline, how this is 2013. So from here to here, the first 5 petabases took 3 years. And then this is about a year for the next one. And then a little bit less than a year for the next one, and then several months. And now you look and it's just growing. So you're sequencing 5 petabases. We're basically now we're sequencing in the range of petabases every month. And so it's just growing really, really fast. So here's just looking at the curve. So we want to compare genomes. We have all these genomes, and how can you compare them? And one simple way is, so if you think about it, you have 300,000 genomes. Well, one easy way to compare them is if you could get a number for them, right? And there's a couple of numbers that make sense. One number is just how long is it? What's the biggest genome? And so this is what I had you guys do a few minutes ago on the web. And you can sort by size here. And 35 megabases. And whoever it was, one of the students said that, but it just says bacteria. That's not very useful. It's 35 megabases, okay? So here are these, and it goes fairly quickly down from 35 to 25, 17. Now, some of these we believe, but some of these are like, man, that's awful big. So we can look at these a little bit more carefully now. So again, this is what we're doing before, looking here at assembly level. And so this guy, if we look this up, this bacteria, we can click on it, and it says on it, anomalous assembly. And the name is just a bacterium. They say it's a full genome, but it's contaminated, they say. And so what are you supposed to do with this? I mean, it's in there, but is it a real? Well, this is done by the University of Zagreb in Hungary. These guys, you know, it's part of the MBL, but they're kind of European Molecular Biology labs and also European Molecular Biology Institute. But these guys aren't that experienced on it, and it's contaminated, and so this is a problem. So if we look at now the next one, the Diamonas, it says that it's full. It looks okay, but it says abnormal gene to sequence ratio, and then it says genome too large. Okay, that's still in there. This is done by the French, by Genoscope. And this is also done by Genoscope, and it's the same thing. It says genome too large. And they have these, and they're not really the size they're supposed to be, okay? And then now we have this next one. It's another bacterium. This is done by the same group in Hungary. And finally, we have an E. coli. This is done by a Danish group. It's actually one of my former students is part of this. And they claim, you know, the GenBank file says that this is like, it's much bigger than here. Actually, I have it here. It's 3,900 records. And, you know, they claim that it's like 20 or 30 million bases long, but E. coli should only be 5 million bases long. And so it's a real problem. And it turns out that it's not one E. coli, but it's actually a collection of E. colis. And so that got in there. So finally, we got this guy, Mumia. And this is from the University of Malaysia. And if you look, everything looks okay on this guy. But then you start looking at, so this is the one, it's 16 megabases. But you look at this Mumia flavor, the same species, and it's only 4 megabases. And so then if you actually you go back and you look, it turns out that just like with E. coli, this guy contains 4 copies, 4 different species, or 4 different strains of the same species, all in the same file. So finally, number 7, now we get down to mini cystitis. And this actually, this is done, whoops. This is a Roseus strain, and I think this looks like it's legit. Yeah, and here's a paper on it. And this actually, so Rolf Mueller, so this guy is the biggest genome so far. And it is, how big was it? It was around 12, 13 megabases. Oh, there it is, 16 megabases. That's the biggest. But you see, it's not so easy. And I did this on purpose. It took a bit of time, but I wanted to show you that this is a problem with big data, is that big data, you get lots of noise in there. And sadly, sometimes you have to manually curate, which is just a pain in the butt. And we're working on, and Liz knows part of this is this DART project, a European Science Foundation grant, on automating data curation so it can throw out all this crap. You know, and so that you, but you know, you have to be careful. You have to know some of the biology. So the point is, when you just look, you say, oh, the biggest bacterial genome is 35 million bases. Well, no, it's probably 16 million bases, and the others are not good. So this is from a group in Braunschweig in Germany, and I worked with these people for many years. This is a good group. So there's 12,000 genes. So this is probably the biggest bacterial genome so far, right? We're still kind of working on it. Okay. And this is a group in Germany, like I said, in Braunschweig. Okay, the smallest genome is the same kind of thing. And I don't know if was this the same one that someone read me here, this guy. So what we can do is we can kind of play around with that. And it turns out that a lot of these are plasmids. They're not actual genomes. And again, you have to kind of, it's one of those things you go through and you look through it. But number 7, Hodgkina, actually is an endosymbiotic genome, and it's only 100,000 bases long. And that's, quote, a true genome. It's greatly reduced, but again, that's kind of a thing. And this is from a group in Montana. And it's a very interesting thing because these are reduced genomes. And Hodgkina, they actually live inside of insects. And they're called endosymbionts, and they diverged quite a long time ago, tens of millions of years, and they've lost a lot of genes because they're living in this nice cushy environment. They're living in the guts of insects, and so they don't have to make a lot of things. So I thought I had some pictures. Oh, it doesn't matter really. Doesn't matter. Maybe I'm thinking of something else. Oh, I know. Okay, never mind. That's coming later in the talk. Okay. So now we look at E. coli. And the biggest E. coli is 17 million bases. This is from the Danish group, and that's not right. And this E. coli is 11, and it's isolated from blood, but it turns out no. So this is actually 4 E. coli genomes, and this is actually 2 E. coli genomes, and this is 2. We're not sure about this, but you know, you look at all these pieces. This is lots and lots of pieces. Now this guy down here is one contiguous piece, and it's 7.6. It's probably real, probably. And then the smallest E. coli you see is only 100,000, and this is not E. coli. And these are plasmids. So this is from a paper published a couple of years ago, looking at the paper is a little bit about different something else, but one of the things we looked at is a bunch of E. coli and salmonella genomes. And so what we did is we looked at the genome quality. And so we shaded it darker if it's good quality, and lighter gray and smaller circles if it's poor quality. And what you can see is that the high quality genomes are around 5 million bases. And they basically have a range from about 4 to 6. And then once you get bigger than that, these are all pretty low quality crap genomes. And similarly, when you get really smaller, they're low quality crap genomes. So you have to kind of be careful about these things. So this is one of the things, is that you have lots of data, but it's a little bit messy. You need to be careful in cleaning it up. So this is a hawk. It's a bird anyway that was outside our window, kind of scaring the other birds away, understandably. So length is one way. It's a very simple way. The other way is you can look at AT content. So how many A's and T's versus G's and C's. And these are looking at different bacterial phyla. And so some phyla are more, they tend to be more AT rich, and some are more GC rich. And some of the phyla, this is part of the characteristics, the genomes are AT rich or GC rich. So we can sort these. And so we can say, okay, what's the most GC rich organism in there? It's 77%, which is pretty high. We look at this column, right? And this guy, it turns out that this lives in soil. And this is basically kind of, yeah, interesting. Now, we can also look at the lowest AT content. And these guys have really, really low AT content. And it's only 13.5 or 86% AT. Now these remember I was talking earlier about these endosymbionts. These guys live inside of insects, and many of them are quite small, 208,000 bases. And so here's the cicadas. You guys know about cicadas, right? Spittlebugs and sharpshooter. And these different bacteria live inside these, and you can look at certain metabolic pathways that are unique to different organisms. And it's kind of an interesting thing. And so if you look at this guy's 208,000 nucleotides long, it's 13% GC. And so it's kind of, yeah, there's a little bit of a side. So you can look at GCAT content, and this affects codon usage, which is one of the first things when you report you can look at. So once you guys have a set of genomes, you need to make a table that says, here's your name of your organism, how long is it, how many base pairs long is it, what is the AT content, and how many genes does it have. This is all kind of standard output for our biotools. And so one of the things you could do then is look at codon usage, and if it's AT rich or GC rich, you're going to see strong bias in the third position. We'll talk about this more in a minute. So you can also look, so if you think about that, length is just how many bases long is it? That's pretty simple, right? Just, the AT content, now you're looking at, is it AT or GC rich? So now you're looking at 2 letters, if you will. And the next one is looking at signatures, where you could look at, for example, 3 letters or 4 letters. You could use 4 letters of tetramers. And these are basically looking at, it's called zero order Markov models, is what the Zoom stands for, but don't worry about that, a little bit technical. But basically, you're looking at how often these tetramers occur. So here's on the side, if you were to blow this up, you could see all the possible tetramers. And you're looking at how they're doing a cluster, you look at how often they occur. And these are a bunch of different bacteria. And you can kind of look at, you can cluster them together. And sometimes you can get useful information. Now, you can also look at not just the ligament composition, but you can look at how it's biased as you go along the chromosome. So this is what's called an atlas, which we'll talk about those later. But here you have your 4 bases, GATC. And what you can see as you go along the chromosome, as you start here, you have lots of G's and A's on this half over here. And on this half, you have lots of T's and C's. G's and A's are purines. And so this particular, this is a bacteria, this is Clostridium tetani. It has G's and A's on the leading strand. So you start here, your origin, you're going this way. At the same time, when you're dividing, it starts in the other strand going that way. So, whoops. So your leading strand has G's and A's, and your lagging strand has C's and T's. And in general, gram-positives. So gram-positives, they have one cell membrane. These are very ancient bacteria. Gram-positive, gram-negative split them out 3 billion years ago. So they're very, very different. They share maybe 5% of the proteins, or less. They're very, very different organisms from each other. Gram-negatives are called gram-negatives because they don't stain. Hans Christian Gram was a Danish microbiologist, and I actually worked with his granddaughter. His granddaughter is the same age as me. And basically what he discovered is that you could add this stain, and some bacteria stain. This is in the 1800s, 1880s, and some don't. And so gram-negative, we still use this today. Gram-negative bacteria are ones that have 2 membranes and they don't stain. And it turns out that gram-negative bacteria, they have a different proofreading enzyme. And then gram-negative bacteria, generally you have G's on one strand and A's on the other. And gram-positive, G's and A's are on the same strand. So in principle, if someone gave you a piece of DNA and you could look at just G's and A's, which bias it is, you can maybe make a stab whether it's a gram-negative or gram-positive bacteria. There are other things as well, but this is kind of a fun article. And so what we basically looked at is a bunch of different organisms and the bias, and sometimes you see very strong, like I was saying. So the gram-positives, the G's and A's are on the same strand, and they have this particular proofreading enzyme called POL-C. And this is from environmental microbiology. We made the cover. That was kind of fun. You could also, another way to compare genomes is you could do alignment. So you could take this genome here, Mycoplasma hypopneumoniae, and you could take another genome and you just line it up. And you can see the red's pretty much lining. Every once in a while, the little blue is things that get inverted. And you have some gaps. So you have some genes here, some regions that are missing in this one. And similarly, you have some genes here that are missing there. So you can line them up that way. And now you can line 2 of them. But this works for comparing a few genomes, but it's not going to scale, right? If you want to compare lots of genomes. You can also generally compare, you can look at repeats, and there's different types of repeats. And so what we're looking at is, we call them local repeats. So you take 100 base pair windows and you're looking within that 100 base pairs for repeats. And if it's 50% AT, you have fairly low number of repeats. But as you go to more AT rich or GC rich, the repeats go up. You get this little smiley face. So I'm going to pick on Andrew since he seems to be talkative a bit. So Andrew, do you have any idea what's going on here? Why would you see more repeats as you go towards 100% AT? So here you have 4 letters, right? GATC. How many letters do you have if you have 100%?
Wait, say that again?
So if it's 50% AT, then what that means is you've got 25% A, 25% T, 25% G, 25% C, right? So you have roughly equal amounts.
A quarter of each, right?
Yep. So you have 4 different bases.
Yes.
Right? Now, if you had something that's 100% AT, how many bases do you have?
2.
You only have 2, you don't have 4, right? So now if you think about repeats, the chance, if you have 4 bases, the chances of a repeat, let's say 3 long, will be 4 times 4 times 4, which is 1 in 64, right?
Mm-hmm.
Now, what is the chance of finding a repeat if you only have 2 letters rather than 4-letter alphabet? Again, looking at 3s. Instead of 4 times 4 times 4, what would it be?
2 times 2.
2 times 2 times 2, right? Which is going to be 8
Mhm.
rather than 64. So what that means is your odds of getting a repeat
Or
are
higher.
much, you're going to have a much greater chance of repeats, right? And so just because
Right.
you're going to a 2-letter alphabet, you're going to see more repeats as you go to AT or GC rich. So these are looking at different types of repeats. So the direct repeats are, they're basically going the same direction on top of each other. Inverted repeats are, they're on the opposite strand. So let's see. So if one's going this way, the other is going to be going like that on the other strand. So, and mirror repeats are, they're on the same strand, just flipped. And everted repeats are, they're going to be like that on the other strand. Anyway, there's 4 possibilities. And so you can look at these and you can look at how often they occur. You can also look at periodicity, so how often things repeat. And so this is looking at fairly short periods. The turn of a helix is around 10 bases, but it turns out it has to do with how it's compacted in chromatin. And in bacteria, it's around 11 bases per turn, the helix on average, roughly. And you can look at these like A's, AA, repeated, and you see this period of around 11. And for archaea, it's around 10. It has to do with how it's compacted. Again, archaea and bacteria are very, very different and ancient. Archaea is kind of from another planet. And so we have this bacteria called Thermatoga maritima, which when we actually look at the genome, and this is, so Karen Nelson sequenced this genome at the J. Craig Venter Institute. So Thermatoga maritima, the proteins, it turns out if you look at the proteins, out of every 4 proteins, 2 of them are bacteria, 1 is archaea and 1 you don't know. And so she gave me this organism and we just looked at periodicity, and we found the same thing looking at periodicity, which is kind of interesting. So the idea is that there's this kind of structural signature looking at periodicity. Peter Warning was a physicist. He's did a postdoc with me. This is kind of fun. So he's more into spectroscopy and looking at periodicity stuff. So this was published 21 years ago. It's a long time ago. But anyway, you can also look at periodicity. You can also, in general, promoters, which are upstream of genes, it's going to be more AT rich than downstream. So if you look at a couple of 100 bases, 200 bases upstream versus 200 bases downstream, it's a little more AT rich. And so in fact, what you could do is you could use this. So because if you think about it, you've got your Tata box, right? It's going to melt. And so this kind of makes sense that it's going to be more upstream. But this brings me, this is kind of related some to your computer exercises. So I think someone was interested in streptomyces or streptococcus. Maybe it's streptococcus, wasn't it? But okay, so streptomyces is a bacteria that lives in the soil, and it's fairly big. It's got 7,700 genes. And what you could do is you could take all those genes and you line them so that they're starting at translation, which is your AUG, where you first that's the first codon when you're making your protein. And so in this alignment, your protein starts here. The gene, the first start codon is here. And so your coding region is here. And then this is upstream. Okay. And actually, let me go back. So this is what it looks like. And I put this shading on just so you can see a little bit better. So what we're looking at is what's called a Z-score. And we're just looking at difference in AT content from the genome average. And you can see that's like, looks a little bit like a seismographer or something, right? You see a little bit of noise, but now if we take a 3-point running average, it's pretty much right on the average for, and then it starts going up. Remember, 200 bases upstream, so here's 200 bases upstream, is more AT rich than 200 bases downstream. And downstream is pretty much the same as a genome because most of the genome for bacteria codes for proteins. It's like 97% coding for this organism. So, okay, so here's your upstream region. Now, when you get to the start, there's this big spike here because there's an AUG. And so they always have an A. That's why the Z score is going to be quite high because there's always an A. AUG, and that's a G down there. And then it kind of goes like this, and then it's going up and down. Now, my question is, what's going on here? Before I call on someone, does someone want to volunteer and explain why this is so different than this? Anybody? So this region here codes for proteins, and this region does not. Now the red line is a 3-point running average. And when I do the average here, it's pretty much 0 because most of the genome codes for proteins, right? So this is, it averages out to 0. And here it averages out to 0. Now, why is this noise so much less than this noise, particularly here, right? So any ideas? I'll give you a bit of a hint that this organism, Streptomyces, it lives in the soil and it's very GC rich. Okay. So, and remember, this is coding. And remember, codons, they're based on triplets, right? AUG is the first codon. And so this is looking at all 7,000 genes now. I've lined them all up in the same direction. And you're seeing this pattern here. So
You're asking why there's difference between like the z-scores once you go over 0 or the distance
Yep.
from translation start.
Yep. And what I'm asking is, so if I do the raw score, it's going up and down, but when I do a 3-point running average, you don't see any difference. It's very little change, right?
Right.
Why the dramatic difference?
That's a good question. I don't know, because don't all bacteria, they don't have the exons, they just have introns. So shouldn't it all be coding regions, right?
This is 100% coding. So I line up all the genes. So this is the coding region. And this is some of the space in between. So this is intergenic here. So this first part is non-coding, but by the time you get further up, because they're fairly close together, you're going to have other genes over here. But so this is all coding, and this is not coding here.
Did it just run into a bigger G? I don't know. Or
So you're looking at differences in A's, looking at AT content, A's and T's differing from the average. Remember I said it's GC rich. So the organism is GC
because
rich.
because codon, because the amino
Yes.
acids all require an A. So whenever you get to a coding region, you're going to have more A's and T's than you are going to have C's and G's.
You're getting there. You're getting there. Okay, flesh that out a bit. How many amino acids are there?
20. 20.
And how many codons are there? Remember, you have AUG, so you have three nucleotides make one amino acid, right?
Right.
So again, assuming random probability, you have 4 letters times 4
So
times
20.
4. No, you have 4 letters. What's 4 times 4?
64.
16.
16, but it's not 2,
16
it's 3 nucleotides. 16 times 4 is what? 16, 16 is 32, 32 and 32 is what?
You mean 32 times 4?
I mean 4
Sorry.
times 4 times 4. What is 4 times 4 times 4?
I think she said it earlier. She said 64.
64, right?
Yeah.
So you have 64 possibilities, right?
Mhm.
But you only have 20 amino acids. So
Yeah.
there's redundancy, right? And do you remember where this redundancy might be? In other words, is it completely random? Or is it in the first base, second base, third base? This is one of the things you're going to look at in your computer exercise.
I know the first one is always methionine.
Well, that's the first amino acid. That's not your first nucleotide.
First nucleotide.
Sorry.
Are you talking about like the cat and then Tada box or no?
I'm talking about, so remember, sorry. So do you remember your genetic code? You have 3. So you have AUG, and then you have maybe ACU and AAA and AGA and GAT. So GAU. So you have threes, right?
Mm-hmm.
So, but if you remember, AUG makes methionine and AUC makes something else.
Right.
Sorry.
Isoleucine. Right. I have a list right here.
Okay. So, well, and here's your genetic code. Now, for
Yes.
Streptomyces, what we've done is there are
They're leucine
7,000. No, well, yeah, a little bit, but there's 6 different ways you can make leucine, right? But it turns out that it's not using all of them equally. Look at this guy. This particular one where you go CUG, it's 158,000 times. This case, UUA is only 156 times. So literally there's 1,000 times preference for this codon over that codon.
Hmm.
So the codons aren't distributed equally at all. Well, so why is this so? So now remember, this organism is GC rich. Okay, so if you're going to make leucine, you have to start with a C or a U. And if they have a choice, they'd rather start with the C because that's a G or a C, right?
Mm-hmm.
And now your second position has to be a U, because this is you're looking at your second position. It has to be a U something, so it has to be CU something, right?
Right.
Now, it could either be U, C, A, or G. Now, if it's a G or a C, that's more GC rich. And look, they occur much more often. If it's an
Right.
A or U, that's less GC rich and they occur less frequently.
Interesting. Cool.
So this is called base wobble, because it turns out for many, so like for proline, C, you need a C and a C, and then you could have any, it doesn't matter what it is, you're still going to get proline. See, all these are proline. Same for threonine, same for alanine, same for valine. So for many amino acids, all you need is the first 2 letters, and that's enough. So the third one's called wobble. So you have 3, when your codons, when you have your tRNA and it's binding, that third position doesn't matter as much. And if it doesn't matter as much, then you can have a bias in there. So you can look at the bias. So one of the things you're going to be doing in your exercise is looking at the bias in the first position, second position, third position. And we can actually plot this looking at AT content. So it turns out that genomes that are very AT rich in the third position, they're going to show a strong bias towards A's and T's. And genomes that are GC rich show strong bias here. And genomes that are 50% are right in the middle. And so a lot of what's happening here when we have codon usage bias, it has to do with GC content. The 2 are kind of related. One's kind of driving the other. And this is a paper we did last year. Well, now I guess 2019. So it's looking at hepatitis, and we can look at codon usage for hepatitis. And so here, now we're looking at codon usage for your 64 codons. These organisms are 50% AT, and you don't really see a strong bias one way or the other. Now, if an organism is GC rich, it's only 25% AT, look at the difference here. See this? So we've arranged it so that the codons that end in C's and G's are below. If they end in A's and T's, are above. So most of the codons, they end in G's and C's. There's a strong bias for organisms that are GC rich. And organisms that are AT rich, what do you think? They're going to be having codons that end in A's and T's. So you can look at this codon usage bias and get an idea for this. So Mike, you're on here still, right? Mike Lexi?
Yes.
Do they make plots like these now? I can't remember, to be honest, in the RBio tools.
Yes, there's a function in RBio tools to I think it's plot usage, and you can specify whether you want to look at amino acids, at codons, or position bias. And so you can do all of these figures that Dave has been showing over the last 5 minutes or so.
So if you're comparing genomes and they differ in GC content, you could look at this and you should see differences. That's the point. Okay. And as Mike said, we could look not only at codon usage. Remember, there's 64 codons, but now we can look at the proteins. We can look at the how often each amino acid is. Now, in general, simple amino acids. So there's aliphatic. So there's glycine, alanine, valine, leucine. These guys, isoleucine. These are abundant. These are just methyl groups. They're really cheap, abundant. They're there. You have your aromatics, phenylalanine, tyrosine, tryptophan. These are more expensive. You see they're much more rare. Histidine. You have your charged here, glycine, arginine. These both have a positive charge. Aspartate and glutamate have negative charge. Asparagine, glutamate, these all have negative charges. Serine, threonine are polar. These have OH groups. You have methionine here, and then you have cysteine. So here you have your two sulfurs. Proline is kind of a helix breaker. So anyway, so you can look around and you can look at this usage, and this something's 50%. You're going to see lots of aliphatics, and you need some charges as well to make things. Now, if you look at something that is GC rich, so on 25% AT, you see a little bit of difference there. And the interesting thing is if you look at arginine, you see kind of more arginine. Here, there's not a whole lot of difference in arginine and lysine. But in both of these guys, you see arginine preferred. And it turns out that's because in order to make arginine, you need more GC-rich codons. And if something is AT-rich, you have a preference of lysine. Because, and if you need a positive charge and you want A's or T's, you go with the lysine codon, where here you go with codons for arginine. So you can kind of look and see. And the other thing is that kind of in neutral conditions, leucines tend to be favored more, leucine alanine in the aliphatics. In the GC-rich organisms, you have alanine favored, and the AT-rich is isoleucine. And again, it has to do with the codons and usage. And so that's just some things you can look. Okay. So we can also look at transfer RNAs. Now you can sort by how many transfer RNAs. And I can't remember who. Someone was interested in Vibrios earlier, they said. And Vibrios, I think, are fantastic organisms. They're very, very successful. They can grow really, really fast. So look at this. You have 297 tRNAs in this genome, in Vibrio parahemolyticus. Okay, so Andrew, how many tRNAs would you need? Minimal tRNAs to live.
Lots.
How many amino acids are there?
20.
20. You need one tRNA for each amino acid, right?
Yes.
Do you need anything more?
No.
No? So ideally, you need 20. That's the middle that you need to live, right?
Yes. Yes, at minimum.
So Vibrio has 297. Why would they need so many? I look down here. Here's some photobacteriums down here as well. So there's a lot of photobacterium here. And so if you look, they have also lots of ribosomal RNAs as well. It turns out that organisms that grow really fast, you need to make lots of protein as well. In order to make protein, you need extra copies. And so this is basically just looking at, and so you can kind of look at these numbers and say, hey, these guys probably grow really fast. And so Vibrio cholera can double in 5 minutes in your blood. 5 minutes to go from 1 Vibrio cholera to 2 Vibrio cholera. And if it's growing exponentially, it goes 2, 4, 8, 16, 32, right? Pretty soon you're going to have, basically you die. Unless you find it treated. Every 5 minutes, that's just phenomenally fast. And so you can kind of look just how many tRNAs you have. Now you can say something about the organism. This probably grows really fast. It can. It's capable of it. So, okay. So you can look at how many tRNAs. Another thing you could look at is small RNAs that don't code for proteins. And this is from a paper, a PNAS paper. It's Shady, one of my students. This is kind of fun. This guy, Jörg Vogel, is an RNA-seq guy. And what we basically what you can do is we took a particular strain of salmonella and we could look for all the small non-coding RNAs, and then we could look at conservation against different organisms. And what you see is in other salmonellas, you can find most of them, but when you start getting a little bit further, even in salmonella, some of them start missing. And now once you get outside Salmonella entericus, or Salmonella typhimurium actually, then you start seeing less conservation. And then by the time you get to, so these guys, it's a little bit trivial, but these are all Salmonella enterica. By the time you get to Salmonella bongeri down here, you see more than half of them are missing now, these small RNAs. And even by the time you get to coli, which coli is very close friends, very little conservation. And by the time you get to other gamma proteobacteria, it's hardly there at all. So the small, these are RNAs that are stable RNAs, but they don't encode proteins, and they're not that conserved. The structures are, but the actual sequences are not. And so now I'm looking at, oh, they're looking at DND. Well, they're looking at substitution. So this is from a talk that I'd given in February for my course. And I put this slide in, and now I'm looking to see, I have no idea where this slide came from. I'll have to look this up, what they're talking about. Mutational frequency and genome size. Deep sea hyperthermophilic anaerobic archaea. Sorry about that. That kind of came as a surprise to me. Obviously I knew about when it came. Okay, I'm not sure what that came from. But so anyway, the idea is that you've got your genomes here, and you have plasmids and other things being added to it, viruses coming in, they get bigger. So you have these regions here that are added, and then things get deleted out and you have genome reduction. You have this kind of equilibrium between them. And so when you're looking at pan and core, you're looking at a combination of what is, so the gray would be the core, and then all the colors would be the pan, the total. And we'll talk more about that in the next lecture. And this is Beatrice the cat enjoying the sun. This is over Sunday. Okay, so we've already kind of we spent a lot of time talking about this, the largest E. coli, smallest bacteria, and how many genomes are drafted.